Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2017. If you are new to my channel, please do like, share and subscribe. And coming to the year December 2017, Part B questions. One of the mechanisms used by bacteria for adaptation to changed environment is altering transcription of the genes. In this regard, which one of the following responses is not found in bacteria? A gene with two different promoters for expression in different conditions, that is found in bacteria. Use of different sigma factors for transcription of gene. Yes, you have got sigma 32, sigma 74 like that, which are used for transcription of gene. Then expression of alternate alpha, beta, beta subunits. That is not correct because only sigma factor varies, not alpha, beta and beta dash subunit. So they have asked for not found in bacteria. So 3 is not found in bacteria. Then next one. A highly specific inhibitor that targets the phosphorylation activity of TF2H is added to an in vitro transcription reaction. Which one of the following step is most likely to be affected? Now TF2H, if it is phosphorylated, the CTD of RNA polymerase II results in conformational change in the overall complex, releasing TF2E and TF2H, thus resulting in promoter clearance. So now the inhibitor targets the phosphorylation activity of TF2H. So which is the step that is most likely to be affected will be promoter clearance. Answer is option 2. Then next one. During eukaryotic protein synthesis, stress condition. Now the stress conditions include viral infection, lack of nutrients, heat shock, etc. Result in activation of specific kinases leading to phosphorylation of key translation initiation factor that inhibits protein synthesis from a large number of cellular mRNA. Which one of the following factors is the target of the kinase? So it will be EIF2 alpha. When it is phosphorylated, it becomes inactive. Now next one. Imagine the following RNA sequence is translated in the mammalian cytosol and mitochondria. So this is the RNA sequence that they have given. Following are some putative peptide sequences. Now in case of cytosol, UGA, this one is the stop codon. In case of mitochondria, AGG is the stop codon. And UGA in mitochondria, it codes for tryptophan. So C for these combinations. So first one AUG, it is methionine, natural. So AUG, AUA, CUG, then comes stop codon. So 3 amino acids will be synthesized. So A, cytosol will be A. Then for mitochondria, this one will be methionine. Then AUA is also methionine. Then UGA is tryptophan. Then AGG is stop codon. So 1, 2, 3, 4, 5. 5 amino acids will be synthesized. So it will be option 2. So the answer will be option 2. So A, 2. Answer is option 2. Then next one. In all organisms, it is critical that replication initiation is tightly controlled to ensure that chromosome number and cell number remain appropriately balanced. Given below are several statements regarding regulation of replication in E. coli. Hemimethylation and sequestration of ORIC, origin of replication, by a protein called SEC-A prevents initiation of replication. So that is correct. They have asked for not true statement. Ability of DNA protein is an important requirement for initiation of replication. That is also correct. The ratio of ADP and ATP is important as high level of ADP is required for initiation. I don't think there is any ADP which is required for initiation of replication. So C is not correct. Recruitment of HDA protein by sliding clamp inhibits ATP hydrolysis required for initiation of replication. That is also not correct. So C and D is not correct and your answer is option 3. Then next one. In E. coli, though DNA polymerase 1 plays an essential role in the replication process, it is not the major polymerase. Everybody knows that. Instead, the enzyme responsible for advancement of replication fork is polymerase 3. From the 4 DNA structures given below, students made several interpretations about the shorter arm being extended by polymerase 1 and or polymerase 3. Which one of the following interpretations below is correct? Now, whether it is DNA polymerase 1 or polymerase 3, they both are template dependent. Then polymerization take place in the 5' 3' direction and it will add nucleotides only on 3' OH group. So first statement, A will be extended by polymerase 3 but not by polymerase 1. So A is this option. There is no 3' OH group here. There is no template. 3' OH group is here, but there is no template over here. So neither polymerase 1 and polymerase 2 will add nucleotides. Then neither B, so A is not correct. Then next one, neither B or C will be extended by either polymerase 1 and polymerase 3. So polymerase, this one, B, 5' PO4, there is no template, there is 3' OH but there is no template there. So it will not be synthesized. Here also 3' OH is on this side, there is no template. So it will not be synthesized by DNA polymerase 1 and DNA polymerase 3. Here it is 5' OH. It adds nucleotide only to 3' OH end. So here also it will not be synthesized. So neither B or C. Now C case. Here 3' OH is there but there is no template. Here also 3' OH is there but no template. So polymerase 1 and polymerase 2 will not be able to synthesize. So option 2 is right answer. C will be extended by both polymerase 1 and polymerase 3. No. D will be extended only by polymerase 1 and not by polymerase 3. Here 3' OH is there but there is no template. Here 3' OH is there but template is present. So both DNA polymerase 1 and DNA polymerase 2 can act in case of D. So the answer here is option 2, neither B nor C will be extended by either polymerase 1 or polymerase 3. Then next question. tRNA genes of a gram-positive bacteria were sequenced. While some of the genes possessed sequence corresponding to the CCA end of tRNA, others did not. Interestingly, even the genes that lacked the sequence corresponding to the CCA end of the tRNA were found to code for a functional tRNA. This is because the CCA end in the tRNA is not essential. It is essential because CCA is the acceptor arm for attaching amino acids. The genes that do not possess sequence corresponding to CCA ends of their tRNA are repaired prior to their transcription. Now they have already told that some of the genes, CCA is lacking. Then how will you repair something which is lacking? So 2 is also not correct. The CCA end is added to the tRNA during their maturation. That is correct. Then the primary transcript of the gene lacking sequence corresponding to the CCA end are subjected to gRNA mediated editing. Now gRNA, it will either insert a U or deletes a U. So that is also not correct. So answer here is option. Then next one. A construct shown below was generated to assess the activity of bidirectional promoter A and B, which has a common regulatory DNA element X. So this is promoter B, this is promoter A, this is the common regulatory DNA element. The construct was used to transform E. coli. The activity of the promoter is recorded in the presence of increasing levels of an E. coli encoded regulatory protein which bind to DNA element X. So this is how increase activity of the reporter gene. So increasing the level of regulatory protein, reporter gene 2, it will be increased, and for reporter gene 1, it will be decreased. The above experiment is repeated in a mutant E. coli with mutation which abolishes binding of the regulatory protein to element X. Which one of the following graphs best depicts the activity of the reporter gene in the mutant strain? So only when regulatory protein bind to the DNA element X, the reporter gene is activated. Now in mutant E. coli, activity of the reporter gene remains as such as it was in the beginning because the mutation abolishes binding of the regulatory protein to the element X. So the answer will be option 4.